Okay, it's 10:45, so we're going to go ahead and start. I appreciate everybody's attendance here and your time, especially considering the busy and packed schedule we have here. So my name is Spencer Schwartz. I represent Canopy Biosciences. Canopy sells a spatial biology platform called CellScape. So what we're going to do in the next 45 minutes or so is I'm going to give a quick introduction to the technology. And then we're going to have Dr. Mara DiMartino come from Weill Cornell and talk about a project that she conducted that used CellScape data in order to take a look at how actually tumor metabolism has an effect on, or can have an effect on, the composition of the immune response. It's pretty cool stuff. So I'll try to keep my end short so we can get to Mara's presentation. All right, maybe scooch this up slightly. Okay. So first, the reason that we're at immunology, or what we're actually kind of promoting at immunology, is we now have the ability to do whole slide imaging with our CellScape platform. So this for us is pretty huge. So this allows us to use archive slides. This allows us to participate in clinical trials, anything where you don't necessarily have to change the prep of your histology sample in order to participate in a study is something that we can do now. So quite literally, anything that you can section and put on a slide, or I think more importantly, anything that is already on a slide, is something we can do this process on. So in addition to that, we've done everything we can to make the window on that slide as big as possible. So it's actually 16 millimeters by 40 millimeters, which is essentially the entire working area of a slide when you consider the label. The other thing we have now is validated and targeted kits that just make the process of getting started in spatial biology that much easier. So the idea here is to give you a set of kind of common markers in order to probe your samples with that kind of makes sense to get the journey started. So again, these aren't massive 50-plex panels or anything like that. I'll get to the reason why in a second here. So I'd like to talk actually about data for my kind of 15 minutes here. So what we're looking at here is a hyperplastic tumor, hyperplastic tonsil. This is 15 millimeters by 9 millimeters. This is also sampled at a rate of 5.5 pixels per micron. So this is very, very dense sampling for this particular kind of application. And we consider all that together. If I take 15,000 by 9,000 times the sampling rate, what I actually have here is a 741 megapixel image. So before we even talk about how many channels, before we talk about the bit depth, this is already a very large data set. But what I would have you consider, though, is it's also a very highly dense and highly utilitarian data set. And if we go through, we used actually Enable Medicine, one of our partners, in order to analyze this piece of tissue, and we can actually locate 1.8 million cells out of this. So my point in kind of starting here is number one, that the CellScape actually does produce a very unique and very high utility data set. And number two, the era of big data is here already. So we already have the tools, we already have the pipelines in order to generate this sort of data set and work with it in a meaningful way. The other data set I want to talk about is really showing off one of the kind of unique features of our particular way of doing spatial biology, which is this idea of reinterrogation. So what we have here is an adenocarcinoma that we sampled using our cyclic multi-immunofluorescence technique, which is essentially staining with low-plex sets of markers, and then we're going to capture that image, capture that data as images, and then we're going to erase that signal only using photo bleaching. So that allows us then to add the next round of signals so that we can build up these, say, 50 plexes as 5, 10, 15, 20, and it massively decreases the complexity of these assays. The other thing it does is let you take a very logical and planned and hypothesis-driven approach when you actually deploy these assays. So if we take a look at this data set, what we've actually done is taken our base immune profiling panel. So we stain with those samples. We then took the sample and put it in the fridge. We took a look at that data and actually realized that there may actually be TLS structures in this. So we then took that same sample back out of the fridge, and then we stained for only the markers that were required in order to make a conclusion positively whether there were TLS structures or there were not. The idea here is, instead of throwing 50 probes at a piece of tissue and hoping that they work, we can logically build these data sets up to 50 markers in the most rational and logical and frankly resource responsible way. So we never waste resources or time staining for markers that we don't need to stain for, And alternatively, we're allowing you to add the markers that actually start answering your question, as opposed to just hoping that the markers that you added do answer your question. Major difference between us and kind of the way that this is being conducted by other technologies in the field. Taking a closer look at the data itself, though. So this is really what I want to get to. So this is what 182 nanometer pixel resolution and also the contrast of HDR allow you to do. So I can tell not just the positivity or the negativity of particular markers, I can actually tell the polarization of that markers. I can start making more deep and more meaningful conclusions about what's going on in this data. I mentioned HDR particularly. So we do HDR when we capture these signals. I will fully admit that not every sample or every marker needs HDR in order to capture it, but I will say it does benefit every single marker. So what it does is actually add contrast to this image. So by capturing a very, very large expression range, we're able to again, generate a very high contrast image. So here what you're looking at is actually, what I'd have you notice perhaps is the very high detail that you see even in a signal like DAPI in the middle. So we can actually make conclusions about, chromatin features and things like that. And again, that comes from the contrast of the image that we're getting. And that is fundamentally what HDR is doing for you on a kind of a day-to-day basis. But the key again is, this is a very data-rich set. And when you start sampling a very high resolution, very high contrast, you can start making very unique conclusions. For instance, if we take a look at turquoise and the B cell on the left, and turquoise rather, I can say for sure that that B cell is interacting with that T cell because I can actually see the protuberances that are actually going into that T cell and interacting with it. So that's a big difference from saying needing 50 markers in order to, hope that you can decide whether or not that interaction is happening or not. I can see it. Same thing when I talk about interactions between these tumor cells and these being T cells on the left. I know that they're interacting because I can actually see the membrane enveloping around that cell. So I don't need, further corollary evidence or anything that's orthogonal in order to do that. I already had that. It's called morphology. So the idea is basically that morphology can actually encode your phenotype. You don't necessarily need 50 markers in order to do that. If you have those additional markers, they're going to give you a much more clear picture of what's going on, though. If we take a look at another example, so these are T cells attacking tumor cells. So T cells particularly, if they're active, if they're healthy, if they're doing their job, are incredibly active. So this is a favorite of live cell imagers, as you can watch these T cells move around, migrate around, migrating clumps of tumor cells, killing them. They move to the next cell, kill that cell, move to the next cell, kill that cell too. So if we encode that, then through our histology sample as morphology, we can see in the upper left here. So I have a T cell in pink that's very stretched out, is trying to interact with these tumor cells. I would suspect, that even without other markers, that tumor cell is active. It's doing its job. When we compare that to the the T cells on the right, so we clearly have a very sad cell here with its nucleus is highly disorganized and it's interacting with two other T cells. Those T cells are balled up though. So I would suspect that those T cells are inactive. Something's suppressing them, maybe immunosuppressing them. What's great about the CellScape then is, number one, I can get this information through morphology. And number two, if I really want to know what's going on there, I can just stain for PD1, stain for PDL1 in the next round and see if that's happening or not. So this is, again, a very powerful technique, a very powerful data set. Last example here. So again, I said not every marker needs HDR, but some of them do benefit from it. So here what we're looking at is some tumor cells in white. They are expressing PD1 in red. And then we have some T cells in green that are massively expressing PD1 in yellow. Excuse me, I misspoke, PDL1 in red. So you can even see just by looking at this picture, you can kind of tell what's going on. So we have T cells that are massively overexpressing PD1 in order to try to overcome this immunosuppression that they're getting from the signal from these tumor cells that are expressing PDL1, or excuse me, PDL1. But so considering HDR particularly, so if we peel away some markers though, so maybe we'll peel away PD1, we'll peel away these canonical markers and just take a look at PDL1. So we have, of course, massive expression of PDL1 that you see on these tumor cells, but you also see expression on those immune cells. So immune cells, too, also produce PDL1, so it keeps them from eating each other. So the question more, precisely is not necessarily whether in a binary fashion these tumor cells do or do not express PDL1. Really, the question is that expression so high that you would consider that a disease state? So that's really what HDR can do for you. By capturing that entire expression range, you're able to rationally place the expression of particular cells within that gamut. We can do that over samples. We can do that over different tumor types, all because of this idea of HDR capture. So it can be a very intriguing kind of addition to your arsenal there. So maybe just wrapping up. So can CellScape give you clustering? Can it support neighborhood analysis? Absolutely. But I'd have you consider that that's where we are now in 2024. But really where we're going is models that can interpret this sort of image. So when you have the morphology encoded, when you have the phenotype encoded as morphology, the behavior encoded as morphology, this is what pathologists do every day. When you have that information and you have a high resolution, high contrast version of that information, you can start powering either through non-artificial intelligence or artificial intelligence, much more powerful methods of just making conclusions about your sample. So I think having these data sets is really going to power your ability to have conversations that are a little bit deeper in the next 2, 3, 4 years than just being able to do cluster analysis. So again, very unique data set. We're very excited to talk to you guys. So we do have a booth. Luckily, I don't even need to note the number because it's under the giant canopy. And with that, I'll kind of yield the rest of my time tomorrow. I appreciate it.
Hi everyone, thanks for coming. I want to thank Canopy for the invitation to present my work today. And as Spencer was saying, we are working on the effect of metabolism on the tumor microenvironment of brain tumors. Particularly, we are studying fatty acid metabolism as an immune evasion mechanism in glioblastoma. I'm a postdoc at Weill Cornell Medicine in the Radiation Oncology Department in Dr. Van Poelbok's lab. And these are my disclosures. As Spencer said, part of this work was supported by Canopy through a pilot award that was really changing the perspective of our results, and you will see why. So this is very, very interesting for us. So we're working in glioblastoma, which is one of the most common primary malignant brain tumors in adults. And the standard of care for these tumors is surgery when it's possible, when the tumor is operable, followed by radiation and chemotherapy. And this is very important to highlight that all of these patients will receive radiation even if the tumor is not operable. And we know from other tumor types that radiation can induce a systemic anti-tumor immunity. But the main problem with glioblastoma, and we think that radiation is not inducing anti-tumor immunity here, is that most of the recurrences you see here, around more than 80% of, you cannot see my pointer, but you see that more than 80% of the recurrences occur within the irradiated field, suggesting that radiation in these tumors is somehow promoting immune evasion mechanisms. And as I mentioned, results from our department and others have demonstrated that radiation can induce systemic anti-tumor immunity by inducing recruitment and activation of dendritic cells and CD8 T cells. However, because of this effect of radiation in an anti-tumor immunity, there are a lot of clinical trials combining radiation with immunotherapy, such as immune checkpoint blockade. And there are some promising results, but in glioblastoma, all of the trials testing the combination of radiation with immunotherapy have failed. I put here 3 examples to show that in different settings, in recurrent setting, even in a diagnosis, these patients do not respond better in the combination with radiation and immunotherapy. All of this is is suggesting that there are mechanisms by which radiation is promoting immune evasion in these tumors. And a possible explanation for this radiation and immune evasion resistance in glioblastoma is the role of the tumor metabolism. And now we know, I didn't see a lot of talks in immunometabolism, but there is this link between tumor metabolism and the anti-tumor immune response. And you can see here in this graph on the right that the byproducts of all the pathways that can be altered in cancer cells, they will have an effect on immune cell function. And also, it's important to highlight that GBM is defined by tumor metabolism alterations. And also, there are some reports showing that these tumors can adapt to metabolic changes in the tumor microenvironment, for example, the changes induced by radiation, in order to survive to these genotoxic stressors. So based on all of these preliminary findings, we ask whether radiation is inducing immune evasion through metabolic adaptation in these tumors. So to test this hypothesis, we ask two main questions. First is whether radiation is inducing metabolic reprogramming in glioblastoma, and then whether this metabolic reprogramming is leading to immune evasion, and this is where Canopy comes in place. So the first part that I'm going to talk about today is this question whether radiation is inducing metabolic reprogramming in these tumors, and this was published last year. So I'm happy to send you the paper if you want to contact me. But I'm going to show you the results that lead on why we're targeting fatty acid metabolism to overcome immunosuppression in these tumors. So to answer this question, we use preclinical glioblastoma models. We have these three models of glioblastoma: CT2A, GL261, and SB28. Most of the results that I'm going to show you today are in CT2A and GL261. These three models, they recapitulate the features of human glioblastoma, but I want to show you that SB28, as you can see, the last one in the H&E sections, is more infiltrative. So it's a bit different compared to the other two, but the three of them are still good models to study the disease. And then, as I mentioned, I'm coming from the radiation oncology department, and radiation in these tumors is particularly relevant. So we're trying our best to recapitulate what is being done in patients in the mouse models. So what we are using is this small animal irradiator. It's called the SARP. And we do a CT scan to localize the area where we injected the tumor cells. So you can see here this white part that you see on the graph on the right, on the picture CT scan on the right, is not actually the tumor, it's the isocenter, the area that we are irradiating. So we use this CT scan to localize the area where we injected the cells and then to do targeted radiation. So we are irradiating a very small area that is 3 by 3. And this is also relevant, I think, for the spatial analysis that we will be doing after. So we are really making sure that we irradiate only the tumor. Of course, the area surrounding, they will receive a scattered dose, but it's not as high as the isocenter. And the type of radiation that we use is this arc radiation. So it's really mimicking what is being done in patients. So you see the mouse is anesthetized on the bed frame, and the collimator is moving, doing this arc. And this is to prevent that all the radiation dose is going in one direction and to minimize the side effects. So using this model, the first thing we did, as I mentioned, is to characterize the metabolic response after radiation. So we used different strategies, and as I mentioned, this is published, so I'm going to go a bit fast through all of these. But one of the main metabolic assays to study metabolism in cells in vitro is seahorse. So we did mitofuel and mitostress assays in irradiated GBM cells in vitro. And we found that radiation is inducing metabolic activity in these cells. We saw an increase in oxygen consumption rate. And when we looked by mitofuel, which pathways are specifically increased after radiation, we could see that glycolysis is not changing. We see an increase in glutamine consumption, but really the highest fold change is observed in the fatty acid oxidation pathway. And to validate whether radiation was affecting this fatty acid metabolism, we did a free fatty acid profile of irradiated CT2A cells in vitro. So you have here the controls. Sorry, I'm keep saying you have here, but you don't see my my pointer. So you see on the graphs on the right, on the heat map, we did 0, 6, and 10 gray, so 2 radiation doses. And we could see that both they increase the abundance of several fatty acids. Particularly, we saw an increase in polyunsaturated fatty acids, and it's all the graphs that you can see down there. And to characterize the effect of radiation on fatty acid metabolism in vivo, we used two spatial strategies. What we did is to inject CT2A cells. We irradiated at day 10 with a single dose of 10 gray, and I will come back to this later because it's important. And 5 days later, we used two spatial lipidomic strategies to characterize the metabolic changes in vivo. We use an untargeted profile using MALDI, and we did a targeted, based on the results that we had from MALDI, we did a targeted approach through DC. What we did is in this section, H and E, to delineate the tumor, our region of interest, and we determine the abundance of each metabolite in this region. And this is just to show you that the main difference between the two techniques is that in MALDI you need to spray the samples with a matrix, and in DC you go directly to the mass spectrometry. So these are the results from MALDI. We did, as I said, we have untreated mice and mice that received 10-gray radiation, and we could see in the PCA plots that the samples are nicely separated. And in the heat map, you can see the metabolites that are affected after radiation. We have a very nice increase in reproducibility between the three samples. And this is just to show you some representative images of some of the metabolites that we saw altered after radiation and the quantification. So based on these findings, we did a targeted approach through DC. And you can see here on the left the H&E. So the control tumor is, of course, bigger, is the one on the top. And the treated tumor is smaller. And this is important because I think, especially for this symposium and for this workshop, that you see that in these tumors, in brain tumors, it's really hard to use other techniques such as flow cytometry, or even we try to do mass spectrometry isolating the tumors, but sometimes the treated tumors are so small that we need special techniques to characterize them, because they are really hard to dissect from the mouse brain. So here what you can see is that a lot of the metabolites that we observed that we found upregulated through MALDI were also modulated, and we could see the same result by DC. So this is an overview of the fatty acid metabolism pathway, and as you can see here, the intracellular fatty acid pool can come from different sources. The main source of de novo fatty acid synthesis is mediated by the fatty acid synthase, or FASN, and this is important because it's going to be the main target for the rest of the project. But you can also have fatty acids coming from intracellular uptake, mediated, for example, by fatty acid transporters such as C36 and fatty acid binding on transporter proteins. All of these fatty acids that are inside the cell, there will be part of the intracellular fatty acid pool, and they can be stored as lipid droplets. So sometimes the accumulation of too many fatty acids can be toxic for the cell, so a survival mechanism is to store them in lipid droplets, and these then can be used to generate energy through fatty acid oxidation in the mitochondria. And as I mentioned before, we saw by in vitro and in vivo in the free fatty acid profiling and also in the spatial lipidomics a particular increase in polyunsaturated fatty acids. So we were also interested in other enzymes such as FATS1 or FATS2 that are the enzymes responsible for the synthesis of this particular class of lipids. So the first thing we did was to see whether radiation was modulating any of these mediators in the fatty acid metabolism pathway. And we found by immunofluorescence in vitro that radiation is inducing the expression of FASN, the fatty acid synthase enzyme responsible for de novo fatty acid synthesis. And we also saw that radiation is inducing the expression of FATS2, the enzyme responsible for PUFA synthesis. And by using the data from the free fatty acid profiling, we could determine the activity of FATS1 and FATS2, and we could see that radiation is also inducing the activity of these enzymes. Finally, as I mentioned, these fatty acids can be stored as lipid droplets, and what we found is that radiation is also inducing accumulation of these lipid droplets by flow cytometry, and also we could detect them by immunofluorescence. To see whether this was also occurring in vivo, as I mentioned, all of this was done in cells in vitro. We used a PET-CT strategy using fluoropropionic acid as a tracer. And this tracer is correlated with FASN activity in different tumor types, such as pancreatic cancer, prostate cancer. So we used this as an indirect measurement that radiation was modulating FASN activity in vivo. So what we did is, again, inject the same cells. We irradiated at day 14. we irradiated tumor-bearing mice, but also normal brains to make sure that radiation was affecting the uptake in the tumor, but not in the normal brain. And 1 day later, we measured the uptake of this tracer. And what you can see here is in the images that there is a nice uptake in the tumor in these models. This uptake is increased with radiation, but is not modulated in the normal brain, suggesting that radiation is affecting the uptake of this compound only in the tumor, but not in the surrounding tissue. Finally, to validate these findings in patient samples, we took advantage of the GLASS consortium dataset, and we selected 55 patient samples that we had paired samples of diagnosis and at recurrence, suggesting that all these patients received at least radiation. As I mentioned, they can also receive chemotherapy. And we did an unbiased approach, selecting all the pathways that include the terms lipids or fatty acid. And you can see in red all the pathways that are upregulated in the recurrent setting. So we can see that most of the pathways that include fatty acid or lipid terminology, they are increased in the recurrent setting, meaning might be after radiation. Of course, there is other factors. And when we looked specifically at the pathway reactor metabolism of lipids, we could see a lot of genes that are upregulated in the recurrent setting that are involved in lipid droplet formation, fatty acid synthesis, and arachidonic acid metabolism, really validating some of our preclinical findings. And finally, if we look one by one at the upregulation of, at the score of the reactant fatty acid pathway in each one of these patients, and you can see in red the match samples, and in most of them you can see that in 39 out of 55 of the patients, this pathway is increased in the recurrent setting. So from this first part, and this is the part that is already published, we could see that radiation is inducing a strong metabolic reprogramming in glioblastoma, specifically towards fatty acid synthesis. And I didn't show everything today, but we showed that most of the metabolic reprogramming is actually mediated by FASN, by this enzyme fatty acid synthase. So we asked then whether this pathway represents an immune evasion mechanism in this tumor. And why we ask this question is because there are many reports showing that fatty acid metabolism can impact the tumor microenvironment at different levels, and all of the pathways involved in fatty acid metabolism, they have an effect on immune cells. You can see that fatty acid uptake, the formation of lipid droplets, the de novo fatty acid synthesis, and fatty acid oxidation, they impact the activation and the function of dendritic cells, lymphocyte T cells, and also regulatory T cells. So based on these preliminary findings and in our preliminary findings and all these reports, and I think this is important to stress out that some of these, a lot of these results were shown in different tumor types, but there are not so many reports on the role of lipid metabolism in glioblastoma. Particularly, the enzyme that we're going to target, FASN, has been shown as an immune evasion mechanism in viral infections. There are a lot of reports on the role of FASN in viral infections, especially now with COVID and everything, but there is nothing about the role of this enzyme in immune evasion in glioblastoma. So based on this, we hypothesize that radiation-induced fatty acid metabolism promotes immune evasion in these tumors. So to characterize to test this hypothesis, this is when Canopy comes in place. We apply to the pilot award. So the award allowed us to analyze 4 samples. And what we did is to inject CT2A cells that had a construct to generate a doxycycline-inducible construct, with a non-targeting sequence, so SH non-silencing, or an shRNA against FASN. 6 days after injection, we induced the expression of this construct with doxycycline by giving doxycycline to the mice in the food. And at day 10, we irradiated again with a single dose of 10 gray. And 7 days later, we looked at the immune infiltrate through Canopy, but also we validated some of these findings by immunofluorescence. And this is just to show you that the knockdown is working very nice. We have here the stain of FASN in the tumors, and we don't see expression of FASN in the FASN knockdown tumors. So here is just to show you the 4 samples that we provided is SH non-silencing untreated, SH non-silencing with 10 gray radiation, SH FASN with 0 gray, or SH FASN with 10 gray. And in the upper pictures, you can see that what they did is to scan the entire brain. And the first part, I forgot to put the lesion, but the first part is autofluorescence, so you can see we can detect the tissue. And then in the lower images, we have CD11B in red and CD45 in white, so we can use this to localize the tumor. You see that the control tumor is the bigger one here on the lower part, and then the treated tumors, either with radiation alone or in combination with FASN inhibitor, they are smaller. And we used this to localize the tumor and do a quantification there. So I'm just going to show you some representative images of this tumor. That is the nicest picture that we have, but then I'm going to show you the quantification for all of them. So again, this is a CT2A tumor where we knocked down the expression of FASN and we irradiated with a single dose of 10 gray. And you can see here in the red square, the area that we selected to show you these pictures. And we used a 16 color panel through CellScape. And I just wanted to show you here that we see a representation of most of the immune cells that are in the panel. And this is very relevant because I know, like, I don't know if you're familiarized with brain tumors. Some people is still skeptical that they are immunoprivileged, but clearly there are a lot of immune cells in these tumors. And this is just to show you that we not only see all the populations represented, but we can also detect, for example, M2 macrophages. So you see expression of CD45, F480, and CD206. We also can detect proliferating immune cells through Ki67. We could see a lot of CD8 T cells in these tumors, and this is important for us, and I will show you later. And we could also detect some Tregs. Here you can see here the expression of CD4 and FOXP3. So based on this, what we did is different analysis, and this part of the analysis was done in collaboration with Matthew Ingalls from Canopy. So we did different analysis types. I'm just gonna focus on CD8 and CD11C cells here because it's really relevant for radiation and to simplify it. But we have done, he did a lot of neighbor analysis and everything. But yeah. So what we did, as I said, in collaboration with Matthew, is to generate these Voronoi plots. So only highlighting non-immune cells in light gray, other immune cells you see in dark gray. And then we have disease in light blue, CD8 T cells in green. We saw particularly a population of CD11C positive cells that they also express F480. And we have Tregs in yellow. As I mentioned, we don't have a lot of Tregs, so it's not so clear in the images, but we could quantify them. And what we did is to quantify these cells and normalize by the area because, as I mentioned, the control tumors are way bigger than the treated tumors. So what we could find is that if you see the bar graphs on the lower part, we saw that radiation is inducing the infiltration of CD11C positive of dendritic cells, that are represented by CD11C positive cells. And this is also true in when we knock down FASN alone and also in combination with radiotherapy. In the second graph, you can see that particularly in the FASN knockdown irradiating tumors, we have a huge increase in this specific population of CD11C positive cells expressing F480. And also in these tumors, we see a huge infiltration of CD8 T cells, and this is very clear, I think, in the Voronoi diagrams with the green cells. And we could also see that in these tumors there is a reduction on Treg infiltration. So based on these findings, we started to study the immune response in these tumors, and we did a validation in the same samples through immunofluorescence. So as I said, we use the exact same samples, SH non-silencing and SH FASN with radiation. And we could validate that FASN knockdown alone or in combination with radiotherapy is inducing infiltration of CD8 T cells. And also that radiation alone, FASN knockdown alone, or the combination of the two is inducing infiltration of CD11C positive cells, presumably disease. But now, based on Canopy's results, we think that they might be macrophages expressing CD11C, or maybe microglia. In the brain we have these resident immune cells that can also present antigens and mount an anti-tumor immune response. So based on the findings that we got through Canopy, we are trying to validate this and expand this population of CD11C expressing myeloid cells in the brain. And finally, so all the results I showed you before, they were done in genetically modified cells. So to move to a more clinically relevant model, what we did is to treat the mice with a pharmacological inhibitor of FASN, so this is systemic inhibition, in combination with a radiation-relevant regimen. As I mentioned before, in all the experiments, we did a single dose of 10 gray, and this is not what is being done in patients. In patients, they do fractionation. Particularly, they do a 30 fraction of 2 gray, so the patients go every day from Monday to Friday to receive a 2 gray irradiation. But there is this regimen that is 6 gray times 5 that is currently being tested in clinical trials in GBM patients in combination with immunotherapy. So we thought that might be relevant for our model to try to stimulate anti-tumor immunity to test this regimen. And also, it's not possible to irradiate mice 30 days in a row because they will die before, because the tumors are so aggressive that they die at day 20. So we had to find the compromise. So we tested this systemic inhibition of FASN with this clinically relevant radiation regimen. And you can see on the survival curves on the right that we didn't see any benefit in the survival of the mice when we treat the mice with a FASN inhibitor. But what we did is, when the mice were dying really at endpoint, we collected the brains and we analyzed the immune infiltrate, and we could detect in these mice, even if they were at endpoint, that we saw a trend through an increase in the infiltration of CD8 and CD11C positive cells. So, as conclusions from this part, we found that radiation therapy is inducing a strong metabolic reprogramming in these tumors, particularly through fatty acid metabolism, specifically fatty acid synthesis. We saw that targeting this pathway holds great promises to reactivate anti-tumor immunity against these tumors, and that I forgot to remove that, but we also think that inhibiting this pathway might sensitize these tumors to immunotherapy, because even we don't see an increase in the survival of these mice, we still see an infiltration of CD8 and CD11C positive cells. So we think that these cells might be exhausted, and this is part of the work we need to do to characterize further the immune response. And as next steps, what we are doing is exactly right now, as we speak, with Canopy. And next week, we're going to send them the samples of these mice treated with 6 gray times 5 and FASN inhibitor systemically. And we are going to look at the immune infiltrate in this context. And I'm also doing at the same time characterization through flow cytometry. But as I mentioned, flow cytometry in these tumors is really hard. So I'm very happy that we are able to do this with Canopy. And we are also doing a characterization of patient samples. We are doing primary brain cultures of human slices of GBM samples with a collaboration with Columbia to validate some of these findings in clinical samples. And finally, I want to thank my lab, so Van Poelbox Lab, Canopy, of course, for the invitation to speak today, and particularly Matthew, that was really fundamental for all the analysis of this data, our collaborators, and financial sources. And I'm happy to take any questions if there is any. Thank you. Yes. Yeah, I think it was 5, but I don't want to.
It's 5. Maybe I'll take that. So the limit is actually hasn't really been found yet. So the tissue integrity that you see is pretty. We've actually had. Trying to think of probably the best example, honestly, is a PBMC sample that's actually just plated on a cover slip. So pretty fragile prep in general. We actually had one of our users run 80 cycles over that, just looking at photo bleaching. Yeah. So yeah. Yeah. So they just did it over time. So the light dose is so low that it doesn't really affect the sample very much. Mhm. We've we found actually that there's a correlation among all these methods as far as what markers need to be stained first versus stained last. Like CD19 is a bear for everybody. CD3 seems to be a bear for everybody, and we're not really outliers there. So it has everything to do with the clone that you're using in the preparation. But yeah, so there's some considerations, but there's nothing specific to our method. Cool. Yes. Yeah. Frozen or FFPE. Yeah, essentially, yeah. Anything that you can prep through a typical histology process is something that we can use. So there's nothing specific about our reagent system or anything like that. Sure. hmm Yeah. Uh, we do. So we actually lately, so we had our own bespoke software when we started, but we found that we were not simply keeping up with the community at large. So we're just going with compatibility with other people that are offering open source software. VisioFarm will work, yeah. So it's compatible with VisioFarm. I'd also say if you haven't tried QPath before, that's really nice. It's open source and easily expandable. We have a partnership with Enable Medicine, which is all cloud based, but that'll take you. It's very low bar to doing that sort of work, and it's also free to academia. Yep. Mhm. For posterity. So the question is, can I use a fused organ? Essentially, can I use a molecular tool in order to do imaging? We can, and we've shown that. We have data doing that. We found though that honestly, what can be a lot more powerful is if you can find an antibody against that driver. So if you're using GFP, if you can find an anti-GFP and use that as an antibody, it's just a matter of multiplying the signal. But we've done both. It's just really it's the processing of the tissue and having that signal make it through that processing that's more difficult than anything. So yeah, if you figured this out in just wide field microscopy, there's no reason why it wouldn't work with our technique. We have data sets where we've at least stained for 105, and 60 of those showed up on a particular sample. So one of the nice things about having that whole slide chamber is now we can take a look at TMAs a lot more easily, so that we can build up those marker classes and show you what you can do with it. Mhm. Yeah, a list of antibodies we absolutely do. So we have clones identified that work and don't work. Yeah, fresh frozen FFPE, even cell suspensions. Awesome. All right. Thanks again for your time, everyone.